Hi everyone, it's top of the hour here, so we can get going. Thank you for joining us on the webinar today. So basically, we've got two speakers, Emily and Sibylla, both at Certara. If you go on to the next slide, Emily, that'd be great. Emily is did her BSc in medical bioscience in Imperial College in London, but part of this work actually relates to work she was doing in AstraZeneca in the oncology DMPK team. So as you can see, since then she's joined as a research associate working at the QS-TS team, and is working on the secondary intelligence software. Sibylla, I'm sure a lot of you online will know her. She is a chemist from the Johann Wolfgang Goethe University, in Frankfurt, and she also received a PhD working in Sweden and at Uppsala University, and there she had a link obviously with AstraZeneca at the time. She's been at Simcyp for quite a while now, since 2007, and is the deputy head of translational science in the DMPK team. So you'll probably know from the amount of publications that she has, she works in quite a broad range of areas, but she does specialize quite a bit in transport, especially non-CYP mediated metabolism as well. And she's really been involved in a lot of the permeability limited models within the simulator as well. So this work today is forming the basis of a paper which came out last year. We've actually been waiting a bit of time to try and organize when we could have this presentation, because it's a really interesting topic looking at food-constituent drug interactions. So I'll just pass over to Emily and Sibylla, who's going to take us through the presentation today. Thank you.
Hello, can you hear me okay?
Yeah, perfect.
Great. As Ollie kindly presented, introduced me, my name is Hee-Sing Jo, but I also go by Emily. I'm currently part of Certara's QS-TS team, and for this scientific webinar, I'd like to present to you on the topic of food-constituent-drug interactions, the influence of quantitative modeling on drug labeling. There you go. And as mentioned previously, this work was done during my time at AstraZeneca as a final year student, and Venkatesh was my project supervisor at the time, and Sibylla also joined into the project later on, since she was, of course, familiar with AstraZeneca, and she was also interested in the perpetrated interactions at the time. And as Ollie mentioned, the result of this project is already published. You can follow it through this link to check it out at your leisure. So to briefly go over the background of looking into herb drug comedication, it stemmed from the idea that natural herbs and foodstuffs are generally seen as healthier by default, generally from the idea that natural sources would also be inherently healthy. And as a result, certain medicinal herb use, such as through supplements, through foods, certain extract oils, are becoming prominent in patient populations, including the oncology population. So here, the WHO defines complementary medicine as broad healthcare practices that are not integrated into the dominant healthcare system. So this includes the use of these herbal and food products. And based on this definition, definition, in the US, up to a third of adults have been identified to use complementary medicine, and in Switzerland, up to 39% of oncology patients have also been identified to use complementary medicine. So just to give you an example of what kind of herbal uses there are, this is a photo of St. John's wort, whose extract is often used as an at-home remedy for depression. There's also turmeric, which I'm sure many people are familiar with, as it's a common ingredient to use in food. But curcumin, which is a compound that's found in turmeric, is also often extracted separately and sold as supplements like these because of their antioxidant properties. However, unfortunately, patients sharing information of their these herbal uses or certain dietary practices are quite uncommon. So the extent of the herb-drug interaction effects and the patients to target to provide this relevant information is quite hard to estimate. So for now, I'd like to focus on grapefruit juice, which I'm sure, again, many people are familiar with. I'm pretty sure you can buy it down from Tesco. And grapefruit is quite infamous in that you have to avoid it during any medication. So this information is taken from the NHS website, and you can see that a very wide variety of patient populations are being affected in terms of they shouldn't be taking any grapefruit or grapefruit juice during their medication. I mean, this includes oncology patients here when they're taking cytotoxic medicines. So first, I wanted to try to identify the knowledge that was available and common features that were also available in terms of the significant interactions perpetrated by herb and food compounds through querying the Washington database of DDI. A moderate amount of data was identified, and you can see that generally, if you have a very specific perpetrator compound in mind that is found in the food source, for example, hyperforin in St. John's wort, then generally the number of sources for that particular DDI can be narrowed down a bit further. What was interesting for myself was that for grapefruit juice, about double the number of sources were identified, just to show you that the extent of grapefruit use is quite common, and these kind of herb-drug interactions is also quite common. And an additional difficulty that I found was that in the database queried that I found, the perpetrator was simply labeled as grapefruit juice. And I'll go a bit more further into detail as to why this adds some uncertainties and variabilities in terms of estimating the exposure of the perpetrator compound. So going into a bit more detail in terms of grapefruit juice and trying to identify the mechanism in which grapefruit juice induces herb-drug interactions, I looked into this paper by Greenblatt et al., where they looked at the comparison between midazolam PK profile by itself and midazolam when it was co-administered with 10 ounces of grapefruit juice. And you can see about a 1.5-fold increase in the Cmax was identified. When this PK profile was overlaid over the effects from 500 milligrams of chlorothromycin, you can see that chlorothromycin actually does recover the extent of grapefruit juice, herb-drug interaction quite well. So by doing this exercise, we can start to narrow down the effects, the mechanism in which grapefruit juice induces interactions. Knowing that midazolam is primarily cleared by CYP3A enzymes, as you can see here, and also knowing that chlorothromycin is a moderately potent inhibitor of CYP3A enzymes, we start to suspect that grapefruit juice is a similarly moderately potent inhibitor for CYP3A metabolizing enzymes. Now moving on to a more detailed analysis of known clinical interactions. These were identified from literature, and this helped in identifying the extent of herb-drug interactions, or HDI, and also identifying some commonalities in significantly affected victim drugs. So with St. John's wort, for example, here you can see a reduction in the exposure in terms of the AUC ratio. And you can see a common feature in the victim drugs were significant metabolism by CYP3A enzymes. So from here we can start to narrow down that St. John's wort might have an inductive property on CYP3A metabolizing enzymes. For curcumin, there were reduction in exposures in the victim drugs, and the victim drugs were also substrates of transporters through which we can start to narrow down curcumin as a inhibitor of these transporters. And similarly with grapefruit, which is under the citrus family, we can start to see commonalities in the victim drugs being affected in being CYP3A metabolizing drugs. So based on these known clinical interaction pairs, we can also start to identify potential victim-perpetrator pairs. And I've narrowed them down to 3 different types of victim-perpetrator pairs, starting with the potential clinical interactions, which I'm sure is of most interest for most people. This is identified by the victim drug, for example, docetaxel here, having a significant metabolism via the inhibitor isoform. So for example, with grapefruit juice as a CYP3A, as a possible CYP3A inhibitor, because docetaxel has a high fraction metabolized by CYP3A enzymes, we can deduce that the effects of this could translate into the clinic. With the theoretical interactions, these victim drugs may be known substrates of, for example, metabolizing enzymes that is expected to interact with the perpetrator, but the extent of the interaction may be unknown or may be more modest. And of course, we have the no expected interactions, where the victim drugs are substrates of certain transporters or metabolizing enzymes that we know have no clinical interactions for, since from previous clinical interaction analysis, these substrates have not shown any changes in the exposure. Based on that knowledge, we can then start to extrapolate to AstraZeneca's own oncology drugs. And there is, based on the metabolizing enzymes for these 3 example drugs that you see here, and the fact that CYP3A enzymes are a part of these 3 drugs' metabolisms, we can start to deduce that herb drug interactions may have an effect in AZ oncology drugs. So based on this, the aim of the project was to help develop a strategy to prioritize herb or food constituent drug interactions to better assess oncology drug clinical risk. So going on to this project further, a more complete picture of the perpetrator mechanisms were gathered from literature analysis for in vitro data, and this includes any competitive and mechanism-based inhibition or induction data, starting from hyperforin, which is a perpetrator compound that is found in St. John's wort. One of the main mechanisms that it induced herb drug interactions with was identified as induction of these CYP3A enzymes, as you see here, but also induction in efflux transporters. With curcumin found in turmeric, but also available as supplements themselves, they were known to have a mechanism-based inhibition of CYP3A enzymes, as you can see here, and also inhibition of efflux transporters like PCRP and PGP. For bergamottin, unsurprisingly, there were also inhibition data, both competitive and mechanism-based inhibition data against CYP3A metabolizing enzymes and other metabolizing enzyme isoforms like CYP2D6, CYP2C19, and so on, and some data on efflux transporter inhibition. So this kind of information was later on compiled and used to build the PBPK model. Now that the main perpetrator mechanism was identified, we now have to move on to looking into the likely exposure scenarios, and we can then compare them with the perpetrator potency. So starting with St. John's wort here, the extract often has these kind of information available where the manufacturer will report the hyperforin concentration. So this is a piece of text that I've taken from a St. John's wort extract that was available commercially. And based on this kind of information from the manufacturer, we can then deduce a likely exposure scenario. So here you're looking at the PK profiles of hyperforin concentrations in different tissues or in circulation based on the manufacturer information. Similarly, with curcumin, as I mentioned, they're available as capsule supplements by themselves. So it's quite easy to use the manufacturer-reported doses by themselves. And based on that, we can also continue to extrapolate the typical exposure scenario for curcumin, as you see here, with concentrations in the systemic circulation or in the jejunum and enterocytes. And we can also start to do an analysis in terms of comparison against the potencies. So now what I want to stress is that estimating the exposure for hyperforin from St. John's wort and from curcumin from the supplemental tablets were generally quite straightforward. You could take the data that was reported by the manufacturer directly, or you could just take the data that was reported as the capsule dose. However, unfortunately, I cannot say the same for grapefruit juice. For grapefruit juice sources, there are very high variability in terms of the grapefruit source that's used itself and also in the preparation method. So for example, certain juices might have pulp that's also included. Some might be 100% pure juice. And this can add variability as there has been literature sources identified that show that pulp, grapefruit pulp, actually has higher concentration of certain perpetrator compounds than in the juice itself. You might also have variabilities and uncertainties from using different kinds of grapefruit, for example, red or white. And of course, there's the preparation method and the strength that the juice is available in. So for example, it could be double or triple strength grapefruit juice that has been diluted down. It's also quite common to see people creating their own grapefruit juice, just blending them together, or just buying a certain branded product. So the point I want to get across is it's much harder to estimate perpetrator concentrations across various different sources, and Sibylla will also go into this a bit more further in her slides later on. Another challenge is having to identify the primary perpetrator that's found in grapefruit juice, and this comes down to a balance of potency and abundancy. So in this table, I've just listed a few candidate perpetrator compounds that I've identified when I was doing this work, and basically it lists the abundancy in grapefruit juice and also its potency against inhibiting CYP3A4 enzymes. And here you can see that we can pretty much rule out bergamottin quite immediately. It has very low abundance in grapefruit juice compared to the other compounds, and also has low potency against CYP3A4 inhibition compared to the other compounds as well. So we can rule bergamottin. Dihydroxybergamottin is a little bit more nuanced, since it is the most potent compound in terms of inhibiting CYP3A4 enzymes. However, the abundancy compared to bergamottin is about 6-fold lower than grapefruit juice, than bergamottin in grapefruit juice. And as a result, bergamottin was selected as the final primary perpetrator that's available in grapefruit juice in inhibiting CYP3A4 enzymes due to its moderate potency against inhibition, but also its high abundancy in grapefruit juice. So now that the perpetrator compound has been identified, we need to continue on in identifying the typical exposure and a typical dose. So a literature search was done on the available data that was known in terms of bergamottin concentration that was measured in certain grapefruit juice sources. And I've identified this one source from Malloa et al., where they used mass spec and to measure the amount of bergamottin that was found in Florida's natural ruby red grapefruit juice. And I thought it was also a good idea to cross-check this certain concentration with another source here from Greenblatt et al., where they measured a similar amount of bergamottin that was reported by their manufacturer in regular strength white grapefruit juice from Florida's Department of Citrus. So this kind of analysis helps me in creating an inclusion criteria for when I need to estimate bergamottin dose, since it is quite uncommon for authors when they do these kind of herb-drug interaction studies to measure a certain perpetrator. And because of the huge variabilities that I've mentioned previously, I need to make sure that when I do select herb-drug interaction studies to use, I need to estimate a consistent, I need to use a consistent source of grapefruit juice so that I can also consistently estimate bergamottin that might be in them. So ideally, I would have a source that would use the exact same type of juice that where we know the bergamottin concentration for, but this also helps creating an inclusion criteria. So for example, having ruby red grapefruit, 100% pure juice, regular strength grapefruit, and so on, so that I can filter down relevant herb-drug interaction studies. So, based on the data that was available in literature and also AstraZeneca's own internal experimental data, I was able to create a PBPK model for bergamottin in grapefruit juice. The details for the input values themselves are much more extensive in the paper, so please feel free to have a look for yourself. Another point that I would like to get across is that while running a simulation, there might be different combinations of a model option that's available for the substrate and the main inhibitor, and in each case, the closest surrogate concentration is used. So for example, if you have a perpetrator compound model, that is expected to have gut interactions, and it also has the ADAM module activated, then the exposure relevant in the relevant interaction site, for example, for efflux transporters, it would be based on the concentration available in the entire site and combined with the data on fraction unbound of the compound in gut enterocytes to get the unbound fraction of that perpetrator. If the ADAM module is turned off, then the enterocyte concentration is replaced with the portal vein concentration. And this is just to show you that by changing the model parameters, then you can also change the driving concentration for the drug-drug interaction or herb-drug interaction. The bergamottin PBPK model established, I conducted model performance against observed data to ensure that the PK profile was being recovered, and you can see generally there is good recovery. What was a bit of a shame was that this particular paper only identified bergamottin concentrations in the plasma up to 5 hours post-dose. Ideally, we would have up to 24 hours to have a more complete picture of the clearance of the compound in circulation. What's also important to note is that for this particular study, they used almost half a liter of grapefruit juice, which isn't really realistic in terms of medication scenarios with certain drugs. So a more realistic exposure would be 200 milliliters of grapefruit juice, which is a little bit less than a full cup. And this would translate to 1.87 milligrams of bergamottin. Now, based on this particular exposure, you can see the predicted concentration time profile here compared with the CYP3A metabolizing enzymes mechanistic-based inhibition potencies. And what I would also like to note here is that bergamottin has very low concentrations in plasma. Just looking at the PK profile here, after 4.4 milligrams of bergamottin from half a liter of grapefruit juice, the Cmax wasn't even able to reach 3 nanograms per mil. Combined with the fact that bergamottin as a compound is highly protein bound with a fraction unbound of 0.018, we can estimate that most of the interactions that's perpetrated by bergamottin would be in the gut. So that's why I've added the gut unbound profile here. Now that the PK profile for the perpetrator has been established, we need to now establish the recovery of the herb-drug interaction effect itself. So to do that, a literature search was done to gather known clinical data with known exposure changes from the herb-drug interaction of interest. Here I would like to just show you a summary of the details of the clinical studies that have been identified that I wanted to use for validation in the future. And for grapefruit juice, you can see that a strict inclusion criteria was applied when looking for relevant clinical studies that use grapefruit juice because of the uncertainties in the bergamottin perpetrator concentration in each grapefruit juice source, as I mentioned previously. So the inclusion criteria was mainly in 100% juice, as well as regular strength, no pulp, and so on. Unfortunately, there weren't any examples of the exact same source that was being used, but generally the close enough source was identified. Now, based on the previous clinical studies, the list of clinical studies that I mentioned, where we knew the changes in the AUC ratio after adding the perpetrator, we've matched the clinical trial demographics, so if it was healthy volunteers or if it were any disease populations, and also the size of the clinical trial in the virtual population. Here you're seeing the geometric mean AUC ratio in the dots here, and the error bars are the 95% confidence interval, and the black dots are the observed from the literature identified previously, and the colored here are the simulated. And you can see for bergamottin and grapefruit juice, hyperforin and St. John's wort, and curcumin found in turmeric, generally the predicted AUC ratio from the perpetrator is being recovered fairly well. Now that this validation has been done, we can then move on to prospective predictions in AstraZeneca's oncology drugs. And the simulation was done in an oncology virtual population just to make it much more physiologically relevant. You can see that from the typical bergamottin dose from grapefruit juice and typical curcumin dose, the extent of the change in AUC ratio is generally quite modest, as in it does not reach more than twofold. However, for hyperforin, a more strong DDI was identified as some of the drug AUC ratio, so for example with osimertinib, would reach to below 0.5-fold changes after hyperforin. However, do note that these are mean ratios, and we've identified that individual AUC ratios, even for bergamottin and for curcumin, they can go beyond over twofold changes in the AUC ratio. So the individual variability is still something that needs to be considered. Now, to account for these vast variabilities, we can then start to do sensitivity analysis. And sensitivity analysis is particularly useful since extreme scenarios can't really be investigated in the clinic. So here, sensitivity analysis was done using bergamottin as a perpetrator to encompass the uncertainty that's found in the individual grapefruit juice consumption, but also in the variability in bergamottin concentration from each grapefruit juice source. And this was done up to 20 milligrams of bergamottin, which translates to 1.5 liters or more than 1.5 liters of single-strength grapefruit juice, which is, of course, unlikely to happen. But this could help encompass more administration of double or triple-strength concentrate use. Now, simvastatin has extensive gut metabolism, as you can see here. And therefore it was predicted to be much more sensitive to bergamottin compared to olaparib, which has higher gut metabolism escape, therefore was selected as a less sensitive, substrate for bergamottin perpetrated effects. And you can see that because of the mechanism of bergamottin inhibition, mainly through the gut, the FG is a driving parameter for sensitivity to bergamottin. And what was interesting is that with simvastatin, with 20 milligrams of bergamottin, about 25-fold change in the AUC ratio was predicted. However, for olaparib, only 1.7-fold increase in the AUC ratio was predicted, so much more modest compared to simvastatin. And what I thought was interesting was that if we go to the drug label for olaparib itself, it says to completely avoid grapefruit and grapefruit juice. However, I'm not sure if this accurately reflects the sort of moderate response that olaparib has to bergamottin. So this goes to show you that depending on the therapeutic index and the exposure response of your victim drug, you could make a more nuanced judgment in allowing herb or food consumptions, or instead of providing this kind of blanket ban. So what have we learned from the exercise? The learnings have been summarized into this decision tree. So first, of course, you would want to characterize your herbal product and also your primary perpetrator. But I do hope I have demonstrated that there are particular challenges in identifying one primary perpetrator, but also in identifying a typical exposure. Of course, you would also want to characterize your victim drug, particularly the mechanisms that would be targeted by your perpetrator, such as any transporter flux data or metabolizing enzyme fractions. And based on the perpetrator and victim drug properties, you can then calculate an R value. And if this R value is below your threshold, then you can already assume low herb-drug interaction risk. However, if this is not the case, then we can start to add more nuance to the decision-making by taking into account the exposure-response relationship. If you don't have the exposure-response relationship, then you can still go ahead with the modeling and simulation activity, such as the sensitivity analysis, just to see to what extent the exposure changes are expected. But if you do have your exposure-response relationship and the herb-drug interaction risk stays below your window, then you can label no herb-drug interaction risk, or you could have a precautionary label in your drug label just to avoid extreme scenarios. If the exposure-response window is reached by your herb-drug interaction, then you have a few options. So one option is to have a precautionary label on your victim drug, of course, but you can also use data from strong CYP modulators, for example, for bergamottin, and extrapolate that data to your herb perpetrator. And this would be particularly useful for sort of worst case scenario predictions. You can also plan a clinical herb-drug interaction study yourself, and this would be if you have the AUC ratio go above a certain safety margin and if there's internally scientific interest to do so. Or you can also just suggest a non-herbal alternative where appropriate. So for example, the general recommendation with grapefruit juice is to just take water. The results of these options can then be fed back into the PBPK model to refine, for example, the perpetrator or victim drug model parameters, but it can also help in extrapolating the effects to other herbs. So for example, with bergamottin, it's also found in Seville oranges, so the work done here could be extrapolated to the effects in Seville oranges, just as an example. So in conclusion, this work presented successful simulations of herb-drug interactions from bergamottin in grapefruit juice, curcumin found in turmeric, and hyperforin in St. John's wort. If you are also interested in the herb-drug interaction effects with the perpetrative mechanism specifically outlined in this presentation, then you can have you can use the models yourself. They're downloadable within the Simcyp members area. Some clear takeaway that I want to get across is the need for standardization when you do herb-drug interaction studies, if that's something you're interested in. So first you have to standardize in terms of identifying a primary perpetrator of interest in your food source, but of course also measure the food source yourself. Generally, it's a good idea to, even if there is a concentration of your perpetrator that has been reported by the manufacturer, just to cross-check that particular source by doing your own measurements. But of course, you also want to standardize to that specific source where you know what the perpetrator concentration is for. Ideally, you would want to standardize everything as much as possible, including the batch. There are also additional enzyme and transporter-based perpetrative mechanisms for the herbs that have been listed here. And if that is a perpetrative mechanism that you are interested in, then it will need more exploration. With that, I would like to thank Venkatesh again for being my project supervisor and Sibylla for her constant support throughout the project. And I would like to hand over to Sibylla now.
Okay, can you hear me? Can you hear me?
Yeah,
anyone?
all good.
Lovely. Okay. Yes, I want to actually continue with the last 2 points that Emily gave, and thank you for this lovely presentation. I really enjoyed it, and I also enjoyed the work together with you and Venkatesh on this. I just want to, in the final slides, point out some additional things. That's first the point that Emily made at the end, that you need to identify the source and maybe additional components that are actually relevant for the interaction, and particularly the concentration, because it sounds so nice if we put it there, but how can we handle that? So I want to go on that. And the second part additional enzymes, transporter that might be involved. Because I know several people that are online have worked with the grapefruit juice interactions, and we all are aware that there are other enzymes as well involved and other transporters involved. However, the set that we had was very consistent, and it's more a standard set. But we know that there are examples which are going out of this set, and I would like to go on these ones. So these are the two things that I would like to go on in the next slide a little bit. So the first is actually this content of the turmeric rhizome, and when you look at that, you can find that the curcumin component is actually described as about 2 to 6% of the complete root. And from this 2 to 6%, it's curcumin, which is actually a ketoenol tautomer, so you have both forms in there. And then you have the dimethoxycurcumin and the dismethylcurcumin. So these are the three curcumins that we are talking about. They are in these 2 to 6%. 2 to 6% can still be a wide range. So what I did, I went to the local supermarket and had a look what is actually available. And this is what is available here. So you had first product, which tells me it has 49 milligram of the turmeric extract. Second has 85 milligram of the turmeric extract. And the last one at 525 milligram of the turmeric extract. And if I all normalize it, I'm getting about 2 to 4% turmeric. So the curcumin is basically in there. So I guess that this is rather from the manufacturer and rather a standard label on there. And also because it's a natural product, it will vary. So I guess it is a good standard label to start with, but at the end, measuring what you really have there is always a good idea. And I think from the clinical studies that have done that, you also look at it. The next one is actually on the grapefruit juice, and that is what Emily pointed out also with a little bit more detail, that obviously with the grapefruit juice you have the issue, that you have the issue that it is different fruits that you can have in, you have different preparations that you have in, and as you can see on this slide, I just combined all different studies that were found for the bergamottin and the dihydroxybergamottin. The lower two lines are basically the ones from the clinical study that Emily showed before. And you see that at the end, you're coming up to 30 micromole bergamottin in there, and about 20% of those is then additional dihydroxybergamottin. So in comparison, so let's say 5 micromole dihydroxybergamottin and about 30 micromole of the bergamottin. And Fukida actually measured a bunch of 8 different white grapefruit juice, and you can see that they ended up with measurements between 10 and about 30 micromole. Interesting, I found that another group, Shirasaka, measured the Tropicana grapefruit juice, which was also measured as Fukada, and they had very different data for the bergamottin and the dihydroxybergamottin. Now, what I found interesting is that it pretty much is the overall amount is the same. It's just a very different distribution, obviously, of that. And in this moment, the dihydroxybergamottin becomes more and more important. However, in the general grapefruit juice, it seems the bergamottin is the driving force, and the driving concentration, and the 6,7 dihydroxybergamottin is not really coming in this high concentrations in at the first part. I highlighted here in three compounds, 3 products that are basically all with about 30 micromole, and these would be the ones I would also choose for clinical studies, and they were very well picked for these clinical studies here. Now, coming to the tricky one, right? Because we know that the standard compounds that are affected by grapefruit juice, and we do the grapefruit juice interaction studies in order to figure out the FG, for instance. And that's normally because it's the 3A4 driven one. We accept that there is interaction with PGP and BCRP, and we can handle that one as well. So I think we have shown that well in the paper. Now, what happens if specific other transporters are involved? Because we definitely have a DDI that shows there is other things also possible, and that's the fexofenadine. Grapefruit juice with fexofenadine, the AUC ratio, if you give it simultaneously, goes down 52%. Or 50 to 60%. So there are two studies. 52% was the Glaser study, CPT in 2007, and 63% was the Dresser study, also in CPT in 2002. The interesting thing that Glaser showed is that if you have 2 hours between the grapefruit juice and the fexofenadine, the decrease goes only down to 38%. And if you have 4 hours in between, there is nothing any longer. So there is definitely something else with the fexofenadine. It was relatively quickly then isolated that it is the intestinal OATPs. And in this case, the OATP 1A2. Now, OATPs in the intestine, I think predominantly they have been neglected, but there are two which we probably in the future will have a look at. It's the OATP2B1 and the OATP1A2. Now, look at means not that I would flag them, because it's a very unique setting, I would say, if you have that. If you find that your compound has interference with OATP1A2, you cannot use the bergamottin file just in order to predict the grapefruit juice interaction. Because in that moment, you need to understand that there is, for instance, also a huge amount of naringenin in the grapefruit juice. And that is already the right concentration to inhibit your OATP1A2. And therefore, you have another component of the grapefruit juice that you need to account for. Unfortunately, we don't really have the naringenin concentration time profile. Ideally, it would be if that's the same concentration time profile as for bergamottin, then we could just add the Ki to the bergamottin. That's not working, okay? So what we do need to do then is understanding how the naringenin is driving it. The difference is also that in that moment, it's not only grapefruit juice, because in that moment also apple juice and other things become, and orange juice become relevant to look at. So therefore, I think it's the huge, if you have a big component of the OATP1A2, then you want to look at it, but it's rather a seldom case as far as I see it. The other one is the OATP2B1. And also there, Shirasaki actually, from Tamai's laboratory, they have looked at it in oocytes transfected with OATP2B1. And they are very aware that for OATPs, you have two binding sites, and they are pH dependent. And they did a very good experiment here because they used actually pH of 6.5, mimicking actually the gradient as you would have that in the intestine. So assuming it would be also working as an uptake transporter, these are the Ki values they obtained. And also there I highlighted in yellow, based on their calculation of their measurement of naringenin in the Tropicana juice of orange, grapefruit and apple, they found that the concentrations in these would be high enough to actually reach this inhibitor concentration or reach this Ki values. So having said that, the OATP2B1, we are still struggling with identifying really the abundance of it in the intestine. Along the intestine, it seems to be pretty much equally expressed along the intestine, but also the location of the OATP2B1. So because it has been discussed also as an apical as well as a apical uptake as well as a basolateral uptake transporter or basal transporter. So if it would be localized in the basolateral side, then obviously you would have a completely different inhibitor concentration you need to calculate with. So therefore, my concern would be, and that has been shown with fexofenadine, only the OATP1A2, and in our data set, that Emily described, we checked actually that none of these compounds would be affected by that. And that's just something that I wanted to bring up, that there are obviously other information here, and I try to summarize all of them here. What I can find in the literature on additional Ki values on additional transporters interfering with grapefruit juice, apple juice, and orange juices. And with that, I actually thank you that you have been patient enough to listen. And now Emily and I are open for any questions.
I can't see any questions coming through here. Is there anyone that wants to unmute and ask any questions? I just had a few questions myself. Probably more relates to Emily's side of the presentation. So obviously, what we're generally considering with grapefruit juice is contaminant administration. I just wondered with the PBPK model whether you looked at kind of the impact of when it's administered or what sort of time frame that you would see the maximum inhibition.
The time of an administration specifically wasn't explored. For this particular work, it was assumed that when patients take oral drugs, instead of taking water to help them go wash it down, they'd be using grapefruit juice. So this would be directly co-medicated at the same time.
Okay.
This is CNS Gusen. Can you hear me?
Yep.
So given that the mechanism of the bergamottin and dihydroxybergamottin is causing time-dependent inactivation, I'm assuming that one would need to dose at least for a few days to get to the maximal effect. Did you consider that?
For dihydroxybergamottin specifically, I don't think there was enough literature data available to make the same kind of PBPK model as I did for bergamottin. But for bergamottin itself, a repeated dose scenario is something that you can explore with the PBPK model that's available in the Simcyp members area as well. But from the just from knowing the most of the interaction will be in the gut and within the system, there's very little that's available. And because most of the interaction will be within the gut enterocytes, unless you're chugging grapefruit juice 1 cup at a time, I think most of the interaction will be quite transient in terms of how long it stays in the gut lumen.
And Tines, just to follow up, from the clinical studies, we mimicked really the clinical studies so that they were matching that one. So as often that was the grapefruit juice given in the clinical study.
Yeah, no, I understand. But I do think for most clinical studies, I mean, the literature, as far as I'm familiar, have shown that the effect probably is a little bit more expressed if one takes multiple doses or a leading dose of the grapefruit juice.
Correct.
Yeah. Okay. Thank you.
I mean, just to follow up, we put these files on the members area that you all have access to it. So, and if you have then experience and feedback to us, that would obviously help to develop that further. So I think it is a way to get a unified compound file together. And one really missing part, as Emily mentioned, is there is not a lot of concentration time profiles for bergamottin in the plasma to actually verify, right? So this is at the moment, it really fits the profile okay. And for the further verification, there were just no more data. Someone is raising the hand, and I have no idea whom. Can someone help?
It was me, Sibylla. It is going back to, what actually was talked about with regard to the mechanism-based inhibition. TNS, it is true that, these effects can, of course, when they are mechanism-based, they can have longer effect. But you have to remember that what defines the, the growth or the turnover in the case of the gut wall is no longer the enzyme. It is the turnover of the enterocyte. We have got models for that which we have published. Therefore, the impact is not as great as you may think with regard to the liver.
Yes, no, I understand that, and I agree. But I do think, I mean, clearly there is some effect where, you're at least killing the enzyme off if you do repeat dosing. But I agree, I understand there would be less than the liver in terms of turnover rate. Those are good points, though. Thank you.
And I mean, just to follow up from that, I would agree to that for all the SIPs and everything. For the transporters, we don't really know yet, right? There are still things open. I mean, you have another question?
No, I tried to turn it off, but apparently I turned it on again.
In terms of the interaction, did you look at any other populations, like just healthy volunteer? Was there any larger interaction than twofold, or
Not
because
from...
in the oncology population, I believe the Schwenger one, the abundances are lower.
Only for the prospective AZ oncology population, I think, required the sort of diseased virtual population. For most of the clinical studies, they were based on healthy volunteers.
Just another comment on this. Again, you should be careful. We are not looking at just on the abundance, because in the disease, particularly, the Crohn disease that, we are working on, as well as, the celiac disease that I believe, sort of some of you working on the Simcyp side, the turnover of the cells are different. And that's going to have an impact with regard to particularly the mechanism-based ones.
Okay. I can't see any more questions here. Yeah, I'd just like to thank Emily and Sibylla for the presentation today. That was a really interesting topic. And yeah, don't forget to find their paper, as well as if you want to, you can download the compound files as well on the members area. So we can leave it like that for today. Thank you, everyone. Thanks for joining us.
Thank you for everyone attending.
Yeah, there is a webinar next week by Felix, and it's going to be looking at PBPK modeling of antibody drug conjugates, if that's of interest to you. And that's able to be registered for on the Certara website. Okay then. Take care, everyone.
Thank you. Bye-bye.
Bye-bye.
Great, thank you.